Now we turn to the matter of how glycogen and starch are catabolized in animals. Both of these, of course, are broken down by amylases in organisms. Alpha amylase is an endoglycosidase and cleaves starch and glycogen chains in the middle, and beta amylase cleaves at the ends of chains typically. Alpha amylase cleaves dietary amylopectin or glycogen to form maltose, maltotriose, and other small oligosaccharides. It's active on either side of a branch point, but the activity is reduced near the branch points. There is a special debranching enzyme that cleaves what are left, and those are referred to as limit dextrins. Note there are two activities of the debranching enzyme. First of all, it transfers trisaccharide groups, and secondly, it cleaves the remaining single glucose units from the main chain. The sites of hydrolysis of starch by alpha and beta amylases are indicated here. The reactions of the debranching enzyme are shown here. Transfer of a group of 3 glucose residues from a limit branch to another branch is followed by cleavage of the bond linking the remaining residue at the branch. It's important to note that metabolism of tissue glycogen is very strongly regulated. Digestive breakdown of starch is unregulated. Nearly 100% of ingested food is absorbed and metabolized in humans. But tissue glycogen is an important energy reservoir, so its breakdown is very carefully controlled. Glycogen consists of granules of high molecular weight. The enzyme glycogen phosphorylase cleaves glucose units from the non-reducing ends of glycogen molecules. And this is a phosphorylasis reaction, not a hydrolysis. Phosphate is the attacking nucleophile. The metabolic advantage is that the product is a sugar phosphate, and that's a potential glycolysis substrate. The reaction of the glycogen phosphorylase is shown here. Sugar units are cleaved from the non-reducing end of a chain to produce glucose 1-phosphate as shown. Note that phosphate is the attacking nucleophile, so this reaction is a phosphorylasis, not a hydrolysis. Now, how is glycogen synthesized? Glucose units are activated for transfer by formation of sugar nucleotides, and that sentence is in bold and red here for a very important reason. It emphasizes that this is really the second time we have mentioned activation of groups for transfer. We talked about activation of carboxyl groups in the pyruvate carboxylase reaction recently, and here's another case. Glucose units have to be activated for transfer to form carbohydrate molecules. The great Argentine biochemist Luis Loar showed in the 1950s that glycogen synthesis depends on sugar nucleotides. Loar worked in very difficult conditions in his laboratory. Argentina was undergoing great political trauma at the time. It was very difficult for him to get supplies. He had very little lab equipment. There's even a story that he built a centrifuge out of an automobile tire. Pretty good story. And he was a dedicated worker who came to work every day in his overalls and brought lunch for his lab workers. He always cooked something overnight and prepared it for his workers so that they could eat lunch in the lab and keep on working. But anyway, Loar discovered the so-called sugar nucleotides and revealed how they are important in carbohydrate metabolism. And for that work, he received the 1970 Nobel Prize in Chemistry. Interestingly, very few chemists knew who Loar was at that time, but he was a giant in the field of carbohydrate metabolism. He showed that UDP glucose pyrophosphorylase catalyzes a phosphoanhydride exchange in a figure we're about to see, and this is driven by pyrophosphate hydrolysis, and that in itself is a very important tag in all of this. We will see over and over in metabolism that release of pyrophosphate stimulates and drives reactions forward because the subsequent spontaneous hydrolysis of the pyrophosphate is energetically sufficient to drive reactions forward that otherwise might not go. Here's a picture of uridine diphospho or uridine diphosphate glucose, either one is fine, and it's abbreviated as UDPG. It's one of the so-called sugar nucleotides discovered by Loar, and it's important to realize that this is an activated form of sugar. It's linked to this phosphoric anhydride in the sugar nucleotide molecule, and cleavage of this bond right here is facilitated by the energetics of the reaction, and we'll see this in just a minute. Here's a picture of uridine diphosphoglucose. UDP glucose is another good name for it. And here's the mechanism of the UDP glucose pyrophosphorylase reaction. It shows attack by a phosphate oxygen of glucose 1-phosphate on the alpha phosphorus of UTP, followed by departure of the pyrophosphate anion. And notice that you've formed another phosphoric anhydride here. You've cleaved one up here, and so the pyrophosphate that's released here, another phosphoric anhydride linkage, can spontaneously hydrolyze to form 2 phosphates, and that is energetically sufficient to drive this entire reaction forward. Otherwise, it would just be an exchange of phosphoric anhydrides, but the subsequent hydrolysis of pyrophosphate is what really drives the reaction, and that produces this sugar nucleotide. This is uridine diphosphoglucose, or UDP glucose. This shows you the structure of a glycogen synthase from a bacterium. It consists of 2 Rossmann folds. Michael Rossmann was a crystallographer at Purdue University, and one of the structures that he studied became famous for its ubiquity in other structures. It was found over and over again in other structures, and this structure came to be known as the Rossmann fold, and it really is this domain here and this domain here. There are 2 Rossmann folds in this structure, separated by a deep cleft that includes the active site, shown here occupied by bound ADP in the purple. Glycogen synthase catalyzes formation of alpha-1,4 glycosidic bonds in glycogen. The very large glycogen particle is built on a single protein called glycogenin at the core. The first glucose is linked to a tyrosine hydroxyl on the glycogenin protein. Sugar units are then added by the action of glycogen synthase, and glycogen synthase then transfers glucosyl units from UDP glucose to the C4-hydroxyl at a non-reducing end of a glycogen strand. So it's important when we look at the mechanism to note the oxonium ion intermediate. We haven't seen one of those yet, but we will in just a bit. Here's the structure of the glycogenin protein. This initiates glycogen synthesis, and the first sugar of a glycogen chain is linked to a tyrosine hydroxyl on this protein. This is the core of all glycogen molecules in your liver and in your muscles. Here's the mechanism of the glycogen synthase. This is an activated sugar, and spontaneous cleavage right here leaves the UDP as a product and produces an oxonium ion intermediate, which can be attacked by the hydroxyl of a glycogen chain to produce the 1 unit longer glycogen molecule. Formation of glycogen branches is carried out by a branching enzyme. 6 or 7 residue segments of a growing glycogen chain are transferred to the C6 hydroxyl group of a glucose residue on the same or a nearby chain. There's a note in your book about what are called advanced glycation end products, or AGEs, AGEs. They're a serious complication of diabetes, as it turns out. Sugars can react non-enzymatically with proteins, and the C1 carbonyl group of glucose forms a Schiff-base linkage with lysine residues on proteins, and these Schiff-base adducts undergo what are referred to as Amadori rearrangements and subsequent oxidation to form what are called glycation end products, and these are irreversibly formed. They do not reverse. The AGEs, the advanced glycation end products, are implicated in circulation and joint and vision problems in diabetics. Measurement of glycated hemoglobin is an ideal diagnostic yardstick for type 2 diabetes and much better than the daily occasional tests for serum glucose. The point is that hemoglobin lasts for about 3 months in your body, and so a measurement of glycated hemoglobin is a pretty good measure of how you've consumed sugars and how many sugars you've consumed over a 3-month period. This is referred to as the A1C test for glycated hemoglobin. All type 2 diabetics are recommended to have an A1C test every 3 months to measure the time average of blood glucose. This shows you the formation of advanced glycation end products. Sugars like glucose can react with the amino group of lysines on proteins to form this Schiff-base linkage, and this rearranges with migration of the carbonyl to the number 2 carbon. So this is really now a fructose linked to the protein through a nitrogen on a lysine, and then rearrangement leads to a whole series of irreversibly glycated proteins. It turns out that there are receptor proteins that actually bind the advanced glycation end products, and these receptors are referred to as RAGE, a receptor for advanced glycation end products. RAGE is a multifunctional protein of the innate immune system that certainly plays other roles in humans, but it plays pivotal roles in diabetes and chronic inflammation and neurodegenerative diseases and T lymphocyte proliferation and cancer because of its ability to bind these advanced glycation end products. It's a single transmembrane segment protein with 3 extracellular domains. The first 2 extracellular domains contain a basic patch and a hydrophobic patch, and binding of ligands depends on interaction of these surfaces, particularly the large basic patch comprised of arginine and lysine side chains. Binding of ligands induces dimerization of RAGE proteins and triggers intracellular responses related to inflammation and tumorigenesis. This shows the structure of 2 of the domains of the RAGE receptor, and you can see the large basic patches right here in the blue, here and here. The large basic patch at the top of the second domain is involved in ligand binding. So now we ask, how is glycogen metabolism controlled? Glycogen metabolism is a highly regulated process, as we've said, and it involves reciprocal control of the glycogen phosphorylase and the glycogen synthase. The glycogen phosphorylase is allosterically activated by AMP and inhibited by ATP and glucose 6-phosphate and caffeine. The glycogen synthase, on the other hand, is stimulated by glucose 6-phosphate. Both of these enzymes are regulated by covalent modification, specifically phosphorylation, as we'll see. The glycogen synthase exists in 2 distinct forms. It has an active dephosphorylated state referred to as GSI, and a less active phosphorylated form referred to as GSD. The phosphorylated form is allosterically activated by glucose 6-phosphate, and there are at least 9 serine residues that are phosphorylated in the formation of GSD. 4 different protein kinases are involved in these reactions, and the dephosphorylation of glycogen synthase is carried out by the phosphoprotein phosphatase 1, also known as PP1. PP1 inactivates glycogen phosphorylase and activates glycogen synthase. It turns out that insulin modulates the action of glycogen synthase in several ways. The binding of insulin to plasma membrane receptors in the liver and muscles triggers protein kinase cascades that stimulate glycogen synthesis. We've seen protein kinase cascades before, and we'll see them again. Insulin's effects include stimulation of lipid synthesis, glycogen synthesis, protein synthesis, glycolysis, and active transport, and inhibition of gluconeogenesis and lipid breakdown. Glucose uptake provides substrates for glycogen synthesis and glucose 6-phosphate, which allosterically activates the otherwise inactive form of glycogen synthase, as we've mentioned. Insulin triggers a series of protein kinases that stimulate glycogen synthesis. Here's a protein kinase cascade here, turning the active GSK3 into a GSK3 that's inactive by phosphorylation. GSK stands for glycogen synthase kinase, and the active glycogen synthase kinase converts glycogen synthase in an active state into an inactive state by phosphorylation, as we've said. So this is the phosphorylation that we talked about in the last slide. Here's the action of PP1, dephosphorylating and activating glycogen synthase. There's also a protein kinase cascade that involves vesicles containing the GLUT4 glucose transporter, and when glucose is imported by this particular transporter, it is phosphorylated to glucose 6-phosphate, which of course exerts allosteric activation on the glycogen synthase D form, the GSD that we talked about. This shows the structure of human GLUT1 in 2 different orientations. It's a glucose transporter similar to the GLUT4 in the previous slide. And this shows the action of insulin on metabolism. As we've noted, insulin binding to its receptor produces protein phosphorylation and second messenger modulation inside the cell. The hormone insulin is considered the first messenger here, and all the agents produced inside are referred to as second messengers, and these modulate a series of different processes in the cell. They activate transport and glycolysis and glycogen synthesis and lipid synthesis and protein synthesis, but they inhibit gluconeogenesis and lipid breakdown. The metabolic effects of insulin, then, are mediated through protein phosphorylation and second messenger modulation. Hormones regulate glycogen synthesis and degradation. Storage and utilization of tissue glycogen and other aspects of carbohydrate metabolism are regulated by a series of hormones, including glucagon, epinephrine, and the glucocorticoids. Insulin is a response to increased blood glucose, and insulin triggers glycogen synthesis when blood glucose rises. Between meals, your blood glucose level is typically 70 to 90 milligrams per deciliter if you're not diabetic, and glucose rises to about 150 milligrams per deciliter after a meal and then returns to normal within 2 to 3 hours. Glucagon and epinephrine stimulate glycogen breakdown. Insulin is secreted from the pancreas, goes to the liver in response to an increase in blood glucose. And it goes via what's called the portal vein, which turns out to be the only vein in the body that feeds an organ. Insulin acts to lower blood glucose rapidly in several ways, stimulating glycogen synthesis and inhibiting glycogen breakdown. This shows the location of the portal vein. It runs from the pancreas to the liver, and again, it's the only vein in the body that feeds an organ. This shows the results of typical glucose tolerance tests for normal and diabetic subjects. Ingestion of glucose at time 0 here raises glucose levels in normal subjects to about 150 milligrams per deciliter, and those levels return to normal after about 3 hours. On the other hand, diabetics typically see glucose levels rise to 250 milligrams per deciliter or more after a meal, and those levels then tend to stay high for hours later. Glucagon and epinephrine stimulate glycogen breakdown and inhibit glycogen synthesis. Glucagon and epinephrine stimulate glycogen breakdown, and that's the opposite effect of insulin. Glucagon is a 29 residue peptide, and it's also secreted by the pancreas. It acts on liver and adipose tissues only. Epinephrine, also known as adrenaline, is released from adrenal glands and acts on your liver and muscles. When either of these hormones binds to its receptor on the outside surface of the cell membrane, a protein kinase cascade amplifies the signal, as we've already seen. This slide shows the actions of glucagon and epinephrine activating a cascade of reactions that stimulate glycogen breakdown and inhibit glycogen synthesis in the liver and muscles, respectively. Here's a close-up of some of that. Glucagon secreted by the liver activates adenylyl cyclase, which produces cyclic AMP, and heart muscle and skeletal muscles can release epinephrine, which also activate adenylyl cyclase. Adenylyl cyclase stimulates the cyclic AMP-dependent protein kinase, also known as pKa, and its activation activates a large number of processes in the cell that lead to glycolysis and gluconeogenesis and glycogen breakdown. You won't be required to memorize all these effects, but you should probably study this slide carefully in advance of taking the MCAT exams. In liver, glucagon typically inhibits glycolysis and stimulates gluconeogenesis. In muscles, on the other hand, epinephrine stimulates glycolysis to provide energy for contraction. So what's the difference really between epinephrine and glucagon? Both of them are glycogenolytic in the liver, that means they cleave glycogen, but they do so for different reasons. Epinephrine is referred to often as the fight-or-flight hormone. It rapidly mobilizes large amounts of energy. It's what makes your muscles tremble a little bit when you see the blue light flashing behind you when you're driving. Glucagon, on the other hand, is for long-term maintenance of steady-state levels of glucose in the blood. It activates glycogen breakdown in a carefully regulated way, and it activates liver gluconeogenesis. Other hormones act in similar ways. Cortisol and glucocorticoids exert a variety of effects on glycogen metabolism. The glucocorticoids are steroid hormones that exert distinct effects on liver, skeletal muscle, and adipose tissue. Cortisol is primarily catabolic. It promotes protein breakdown and decreases protein synthesis in skeletal muscle. In liver, on the other hand, cortisol stimulates gluconeogenesis and increases glycogen synthesis in several ways. It stimulates expression of genes for the gluconeogenic enzymes, activates enzymes of amino acid metabolism, and stimulates the urea cycle, which we'll meet in Chapter 25. Some of these effects are shown here. Cortisol can act on the urea cycle, on the amino acid metabolizing enzymes, on the gluconeogenesis process, and also in glycogen synthesis, activating all of these. It's a very interesting set of slides here that show the carbohydrate utilization during exercise, and we'll take each of these in turn. The frame A shows contributions of the various energy sources in muscle activity during mild exercise. Frame B that we'll look at shows consumption of glycogen stores in fast-twitch muscles during light, moderate, and heavy exercise. And frame C shows the rate of glycogen replenishment following exhaustive exercise. Let's take a look. This shows what happens when you exercise and the percent of total energy that you use in exercise that come from different sources. You see that the amount of energy that you have from ATP rapidly declines. You consume most of the ATP that you have available during the first few seconds of active exercise. On the other hand, phosphocreatine provides a store of new ATP for your body, and phosphocreatine conversion to ATP lasts for about 30 seconds. On the other hand, anaerobic metabolism produces more energy that you can use during the first minute or two of exercise, but aerobic metabolism has to kick in after that and carry out the TCA cycle and produce energy for you. The anaerobic metabolism referred to here is mainly glycolysis. This shows the consumption of glycogen in your body during light, moderate, and heavy exercise. Light exercise doesn't change your glycogen levels too much, but moderate to heavy exercise consumes glycogen rapidly. Imagine soccer players in a soccer match. Soccer matches typically last for about 2 hours, and notice that after 2 hours, even in moderate exercise, all the glycogen content of liver and muscles has been consumed. You'll notice that players don't have legs anymore. They say they've lost their legs. What they've really lost is their glycogen, and they're unable to provide energy for muscle contraction after an intense soccer match. So how is glycogen regenerated, and what are the consequences of diet for regeneration of glycogen? Notice that if you consume all your glycogen, drop it to 0 here after, say, a very intensive soccer match, that a fat and protein diet will build up glycogen very, very slowly, almost equivalent to having no food at all. On the other hand, a high-carb diet replenishes glycogen over a period of a couple of days. So this is why carbohydrate loading, as it's called after an intense athletic event, is a good thing to do. You can regenerate your glycogen quickly in a high carbohydrate diet.